This is a rather, it's not the, it's an unconventional set of ideas, but I hope you'll find them interesting. And it's physics and information, but it's not the kind of Shannon information or entropic kind of information you're used to, but something different called algorithmic information. And I want to tell you a little bit, before I do, just talk about myself a little bit, since this is a chance to humanize myself to you a little bit, and I don't normally get to do that, especially not this last year. So how did I get to where I am now? There's a question somebody asked me. This is me as a child in Gabon in Central Africa, where I grew up. My parents are biologists, and this is Berche, my pet chimpanzee. If you actually, I did a little documentary series on a bunch of big questions in science, where among others we discussed with Jane Goodall whether it was a good or bad idea to have chimpanzees at home. It's actually a bad idea, but it's a long story. You can see that online. Now, my chimpanzee and I were very close friends. We were about the same age. And although we looked differently, at least I hope you think we looked differently, my mother told us we behaved remarkably similarly. And so for a long time, I used to wonder, and I still wonder, what makes us different? What makes a chimpanzee different from me? And these are really the kind of big questions of biology. What makes biology what it is? Well, you might naively think that it's something linked to our genome. And indeed, we've got, we share genes with organisms, and the closer the organisms are to us, the more we share. So we share about 15% of our genes with E. coli bacteria, about 50% with frogs, 50 with flies, 70% with frogs. If you kiss one, it becomes a prince, then that's closer to 100%. And we share about 98% of our genes with chimpanzees. Although we are similar, it feels like we're a little bit more different than that. And the other interesting thing is that the number of genes that we have in our body doesn't correlate very well with our complexity. There are very simple little organisms, including rice, that has more genes than we do, and yet we wouldn't say that we are less complicated than rice. And so one of the things that's emerged over the last few decades is the understanding of how these genes work. And there's a subset of them that work actually not as protein encoders, but as switches primarily. So here I have a little diagram from a physicist called Uri Alom, who mapped out the switch genes in the E. coli bacterium. So each of these little dots is a gene, but each line is a locus of control. It tells you this gene turns this off or that on. You'll see that, for example, this one right here is a kind of hub. It's very important. It turns a lot of things on or a lot of things off. And it turns out that the complexity, the biological complexity that makes us who we are, is linked not just to the number of genes that we have, but also how they're interconnected. And just as you can take a bunch of electronic components and rewire them to make an oscillator or a switch or a wide range of other types of electronic behavior with the same components, so also with biological components, we can reorganize them to do particular kinds of behavior. And the reason why physicists are interested in this is because you look at this thing, it's very complicated. And I'll argue to you in a minute, I'll come back to this and argue that the number of ways that you could connect these is so large that you can't possibly search that space. And yet we seem to be able to find things in that space, which is very fascinating. This tells us that there's some kind of emergent properties in there. There's some kind of lower dimensional order that we should be able to find. And that's where I think some of our ideas from physics will help us to understand the physics of life. So I've been very interested in this question. What is the physics of life? And I'm going to touch on one aspect of it, which is going to link a little bit to this question of large numbers of possibilities. And so another kind of thing I work on in my lab is other kind of projects that we do is on self-assembly. So here is an example of virus self-assembly. So viruses are made of proteins that make little balls, basically, with DNA or RNA inside. So the coronavirus, for example, is an RNA virus, which has a little ball of RNA. And it goes into your cells, and then it makes copies of itself, multiplies by hijacking your cellular machinery. And so it then has to form into newly formed viruses. And so that it makes the proteins, they float around, and they must spontaneously self-assemble. So here you see an example when some of my DPhil students here in Oxford, actually, Ian Johnston, who's now in University of Bergen in Norway. And this is a simulation he did of how these things self-assemble into really well-formed little capsid balls. This is something that's happening if you have any virus at the moment inside your body many, many times. And so it's kind of interesting to understand these kinds of questions too. I was going to say that it's random motion that nevertheless gives you order at the end. So Here's another theme that we're going to explore today a little bit. How does randomness generate order? And I always like to say to people that, science is definitely fun. Here's what I'm doing there with a self-assembly is a bit like taking Lego blocks and then putting some glue on them, shaking them around, and out comes a fully formed train. And if I could figure out how to do that, I would make a lot of money in the toy markets, this is for sure. But I think one of the great things about science is that science is fun. And I got interested in this self-assembly problem probably about, actually well over 10 years ago, because somebody explained to me that this happened, and I hadn't ever realized it. And I thought, well, that's strange, because if I take a bunch of Legos, add glue to them, shake them around, I'm going to get junk, because the vast majority of configurations are junk, because those are high entropy configurations. There's only a small number of well-ordered configurations like this train. So how does this system win over this enormous amount of potential entropy that there might be? That's the kind of question that I was interested in. And you'll see that theme coming back again in different ways in this talk, in a slightly different way. Now, I told you several times now that the number of possible states of these systems is very, very large. And actually, this exponential growth of states is an old idea. In fact, you can find this all the way back down to the Babylonians, who had little games of doubling. And another example of this is the invention of chess. This is a picture from Shamanah, the Book of Kings, from the great Persian poet Ferdowsi. But the story is that the inventor of chess, Sasa, comes to the king, chosen this game. The king is duly impressed and asks Sasa what he wants as a reward for this great entertainment that he's brought. And Sasa says, well, I would like to have 1 barley corn on the first square, 2 on the second, 4 on the third, 8, etc., and just doubling every time. And the story is that the king was at first offended because he thought his great generosity was not being recognized, until his accountants added it all up, and it would be many, many times the world's current grain production. I think 18 quintillion tons is my estimate of how much grain that would be. Something unimaginably large, just on a chessboard. So this tells you that these kinds of number of possible states in all these kinds of systems grows exponentially large. Here's another one of my favorite examples. Yeah, 18 quintillion, I remember it right. From the great Argentine poet Borges. Borges has a really lovely little book, or actually it's more of an essay, called The Library of Babel, where he talks about this magical library, which has every single book made up of 410 pages. Each page has 40 lines with 80 characters on it, using a 22-letter alphabet, along with a period, space, and a comma, so 25 characters. It says every single book exists in one copy in the library. Every possible book is there. In the story, people go into the library looking for what they call their vindication. And the idea is that they're going to find a book that will describe their life. And indeed, your life would be described in this library, your life up to now, and including a book which would predict your life in the future. Borges points out, of course, that book will be there, but so will be all kinds of treacherous variations of that book. And so every possible book you can imagine, every possible life history will be in there. There's a book in there that tells the story of my life up to now, and there's also a book which tells the story of my life up to now. At this very moment, the meteorite comes through my kitchen roof, and I'm obliterated. All those stories are in there. So although every book is equally likely, the question you might ask is every story equally likely? Well, that is probably not true, that they're all equally likely, because some stories are relatively simple to tell, and so they could be told in many different ways, and so they'll be there in many different versions of them, but be all effectively the same story. Some stories are very complex, and you really would need all 410 pages to describe them, and so they'll be there in a much smaller number of copies. And so this is an interesting thing to think about. What are these possibility spaces like? Well, they're hyper-astronomically large. They're larger than anything you could possibly imagine. They contain all possibilities, but some possibilities are going to be more likely than others. And this is the next theme that I'm going to explore with you in this talk, because this question turns out to be very important in biological systems. So you may remember from your secondary school that 2 very important biomolecules are DNA or RNA, which have an alphabet of 4 letters, or proteins, which have an alphabet of 20 letters. So if I'll take proteins as an example, if I made every protein of length L, which is a certain length, then I've got 20 amino acids, so I have 20 to the L possible different sequences I could make. If I made every length 58 protein, it would weigh more than the visible universe. And for RNA or DNA, if I made every length 126 strands, it would weigh more than the observable universe. I've actually written a paper on this called, I call these hyper-astronomical numbers, because you see we're quickly grabbing at metaphors that are linked to the size of our universe. So this is interesting because this tells us the possibility spaces are extremely large, and clearly nature has only searched an incredibly small fraction of these. The average length of proteins in your body is 476, and if I made every length protein of just that length, it would weigh 10 to the 500 times the mass of the observable universe. So these are unimaginably large numbers. If you like numerology, I don't recommend it, but 10 to the 500 is one of the estimates for the number of vacua in the Calabi-Yau manifold. It just could be the number of multiverses that there are. So that's interesting, there's a connection, but it is genuinely meaningless, but fun to think about. So these numbers are so large that even if you made every possible universe, you still couldn't make every possible combination of proteins in them. So this is actually a fascinating kind of question. How does nature work in these extremely large spaces? And I got interested in this question because somebody pointed out, actually a friend of mine in Cambridge, Simon Cohen-Morris, who's a very great paleontologist, I was talking to him one day and he pointed out to me that this experiment by Jack Szostak, a Nobel Prize winner at Harvard, who did an experiment which is in vitro evolution. So you do evolution in a little test tube, and he used RNA structures. So RNAs are these straight sequence of molecules. They can be information carriers, as you probably learned about them in school, but they can also do other things. They can be active like proteins. They can be catalysts, for example, and they'll fold into this three-dimensional structure. So here's a particular structure that's a catalyst. And Szostak was looking for self-cleaving enzymatic activity. So RNAs that will basically catalytically cut themselves. And he puts a bunch of random sequences in a test tube. He picks out the ones that have a little bit of activity and then enriches those and randomizes them again. And you keep doing that again and again. It's like a variation natural selection. And it's a very nice technique, and it works. So every time he did this, he found a good catalyst. But every time he looked at the structure of it, he found the same structure called the hammerhead ribosome. I remember I looked at this and I did a little back of the envelope calculation. So how much material did he use? Well, probably on the order of 1/100 of a gram of material in total in his experiment. And in spite of that, he was able to find the same structure again and again and again. Well, if you measure how much all length 55, which is roughly the length that he used, would weigh, that would be about 10 to 10 kilos. So this is a little bit like having a giant haystack, and you put your hand in, and every single time you put your hand in, you grab a needle. But if that happens every time, it's going to tell you that there are going to be a lot of needles in that haystack. And that's strange. Why does this particular structure appear every time, even though the number of sequences is hyper-astronomically large? That is very surprising. What is nature doing to keep finding the same thing, given that it can't randomly search through the whole space? But as far as we can tell, this process is entirely random. So what makes the outcome of this highly random process non-random? That's the kind of question that I'm going to try to think about today. And one of the themes of my work has been thinking about how random processes have non-random outcomes. So I started DPhil students about 10 years, a little over 10 years ago, working on this. And so the nice thing about the RNAs is that you can take this sequence. Here's a sequence of 4 letters of length 55. You can fold them so you find the free energy minimum that gives you a particular shape. So this sequence actually folds into this particular structure. And although the sequence to structure problem is very hard to solve for 3D structure, you can do a simpler problem, which is looking at the sequence and asking what binds to what. So who binds to whom? And it's called a secondary structure. This is a little depiction of it. It tells you basically that in this sequence, this G and this G on the outside, this A and that U have bound to each other. There's a few binding rules that they have. And this structure is the free energy minimum. And then you can ask yourself questions like, how many sequences will give you this particular structure? Because the binding rules are relatively simple, you can switch some things around and you'll get the same free energy minimum. And so you can ask yourself, well, is there some redundancy in there? And for that, we looked at a simpler problem. So this is a length 15 RNA, so it's length 15. The binding rules first order that G binds to C, and G binds to U, and A binds to U. So this is a sequence, and just if I walk along it from, say, I start with G, G, C, G, G, so the first four are bound, those green things are bounds, then A, these next ones are not bound, and then these are their complementary ones that they bind to, the last two are not bound. I can depict that by the simplified description. So a bracket means I'm bound, bracket, 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 then I've got some dots, dot, 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 dots, and then close bracket, close bracket, close bracket, and these things tell you what they close with, and then some dots at the end. So I can abstract out this structure into this little set of dots and brackets. That's kind of cool. I've turned this into a kind of a digital sequence. And then I can ask questions like, if I looked at every possible length 15 sequence, so 4 to the 15 is 2 to the 30, 2 to the 10 is 1,000, so 2 to the 30 is basically a billion. There's a billion sequences. That's a perfectly solvable problem. I can fold all of them. How many structures will I find? And will I find them all with kind of equal likelihood, or will I find some with higher likelihood than others? Well, it turns out that there's only 431 structures that you find. So even though there's a billion sequences, I've only got 431 structures. That's good, because that means I've reduced the space that I'm trying to look in. But what we find is these first 26 that I show you here take up 50% of the space. So this one has 11 million that give it as a ground state. This is about 10 million. And so actually with a 50% chance, I'm going to get one of these 26 structures if I just randomly pick a sequence. So this is helping me a little bit with understanding the Shostak experiment, because he randomized some sequences and he finds a certain structure again and again and again. Well, that must be one of these common structures, structures that are easy to find. Whereas this structure down here does have two sequences it has as its minimum, but it's going to be highly unlikely. So it's basically a 2 in a billion chance that you'll get this structure by randomly sampling. Whereas this one, you've got a reasonable chance of getting it by random sampling. So that gives you some sense, by 1% chance. So this gives you a sense of that these outcomes are highly non-random. So we worked a lot on this. We looked at length 55, which, just to remind you, was the length that Shostak did. And indeed, what we found was this hammerhead ribosome structure that he finds is one of these structures that has many sequences mapping to them. And you can, if you remember your stat mech, having a lot of sequences mapping to you is like being a high entropy state. So you've got a lot of microstates that map to you. And what this really is a kind of entropic argument, saying the entropy of these different macro states, which are the folded structures, varies over many, many orders of magnitude. And so if I just randomly pick sequences, I'm going to, my free energies, I'm going to be dominated by my S, my entropy term. And that's what I'm going to see, and see a high entropy structure with many sequences. And so here with these green lines, here what we have is this, the neutral set size, which is the number of sequences that map to a structure. Here we've ranked them from 1 to 10 to the 13, roughly, which is the number of structures we think that there are. And you see there's a small number of structures, a very tiny fraction of them, that have very many sequences mapping to them. And there are lots which have an average number, and then just a few with very few. And we looked at all 504 natural RNA sequences that have been found in all across nature that are known to fold, where folding is known to be important. And every single one of them was one of these high entropy sequences, high entropy structures, sorry. And so this is interesting. This is telling us that even though nature has this very large set of possibilities, many different, it's a bit like many different books in the Borges library, many different stories in the Borges library, only the most frequent ones are the ones that are found in nature. And so the reason why Shostak found the same structure again and again is not because there may not be other good catalysts in the space of all possibilities. It's because you're only really looking at a tiny fraction of the set of all possible structures. And that's kind of cool. It's also interesting because this is what's called a non-adaptive explanation. So I haven't invoked any natural selection. I've only looked at the rate at which variation arises. And it's not often looked at in the biological literature. And so what we've done further since then is looked at some simplified structures. So here's another one of these folded structures. You can kind of look at this and you can see, well, I could probably abstract this out in some kind of more coarse-grained level where I take from each of these little arms and make them into just a single. I just look at, like, for example, the number of loops in there. And there's a whole series of ways you can abstract these out and in different levels. And so this structure here has this at level 1, level 2, level 3, level 4, level 5. The nice thing about this is by doing so, I've got fewer structures to look at. And so I might by looking at natural structures, find certain structures more frequently. And then we found something very recently, something very surprising. This is one of my former DPhil students who's now professor in Kuwait. So he pointed out that we could look at this coarse graining of the structures, and then we looked at just randomly sampling sequences. So I just pick sequences at random, see what they fold in, see what structure they give, and I rank them. So for this length 55 at this level 3, which is this intermediate level structure here, we estimate that there's about 10 million possible structures of length 55. And then we randomly sample sequences, and then we rank the structures by how frequently they appear. And what you see is that this drops down by many orders of magnitude. And what's interesting is all the natural sequences that we found there's only 28 of them, are all in this very small subset of highly frequent ones. So although there's 10 million possible structures, nature only uses 28 of them, and we can more or less predict which 28 of those you're going to find just by telling you these are the ones that are easy to find. This is a kind of surprising result, because we tend to think about biological features as being there because they were selected for. So why does this length 55 RNA have this particular structure? Well, because this structure was selected. And here we're saying that may be true, but actually selection is only working in a very small subset of possible structures. There's many more structures that could be possible if you waited forever, but you can't. And so you're only really finding this very small subset of them. And so we can predict some of their properties while ignoring natural selection entirely. So that's the kind of story there. So what I'm just showing you in this one system is that there's a very strong bias towards a subset of structures. And the big question then is, why is this true? Why is there such a strong bias? And for that, I want to take a little detour into information theory. So you probably, or you should have at some point seen Shannon information. So Claude Shannon, the great father of information theory, wrote a paper, which I just recently looked up, has 83,000 citations on Google Scholar, which is unbelievable, must be one of the most cited papers ever. Where in 1948, he basically came up with this formula here for the Shannon entropy of a random variable, so a stochastic variable. And what it basically is telling you is it's trying to capture the information in it, which is why he's considered to be the father of information theory, or the surprise, the uncertainty in a variable. So if I have a coin that's a fair coin, heads and tails are equal to each other, both 50%, then the entropy in bits, so the probability of getting x being plus half, so being up, heads, sorry, is 1/2. If I take this in base 2, so that's bits, then the log of a half is just 1. So I get minus, the log of 1/2 is minus 1. This is actually a mistake here. 1/2 times minus 1, minus 1/2 times minus 1 is 1. That should be 1. And so this tells you that entropy for an unbiased coin is 1, whereas it's not hard to see the entropy for a more biased coin is going to be less than 1. That's because there's a little bit less surprise in what comes out if the coin is biased. And there's a very deep link between Shannon entropy and Gibbs entropy. And in fact, Shannon put in H there because of a link to Boltzmann's H theorem. So the idea of information and entropy are very closely linked. And there's a very active field of work now looking at quantum information in physics more generally. And almost all of this is based really on Shannon information. There is, however, a different kind of information theory, not entirely different. It is entirely different, although there are deep links between the two, which is called algorithmic information. And this is something that was thought up a bit later in the 60s by a great Russian mathematician Kolmogorov and Greg Chaitin and a few others. And they were thinking about a different question, not the entropy of a stochastic variable, but the entropy of a string of digits. And the thing they were trying to capture was something like this. If I have a string like this one, which is 0, 1, 50 times, so a 100 length string of zeros and ones, intuitively this seems simple. And the reason it seems simple is because I could generate it with a simple program like print 0, 1, 50 times. Whereas this string below it, as far as I can tell, is truly random. There is no simpler description than just print the string. And so they said, well, they formalized this by using what's called a universal Turing machine, which is a computer that can do any kind of computation, and saying, well, the Kolmogorov complexity of a string is the length of the shortest program that will generate that string. So this one, print 0, 1, has a smaller Kolmogorov complexity than this one, which is print the whole string. And because a universal Turing machine can do any calculation, it turns out that this definition is actually independent of which universal Turing machine you use, because you can always use a compiler to go from one machine to the other. So up to compiler terms, which are called order 1 terms, terms that are independent of the sequences, you can define a Kolmogorov complexity of these sequences. Now, the interesting thing about Kolmogorov complexity is that you can prove that there exists no algorithm that will give you the Kolmogorov complexity. This is because you can link it to the Turing's halting problem, which in turn can be linked to Gödel's undecidability theorem. So it's kind of an interesting thing. It's a Kolmogorov complexity is an uncomputable object, but it is a real object. And intuitively, why is this hard? Well, if I give you this sequence down below, okay, it's also, as far as we can tell, random. It's actually the first roughly 100 binary digits of pi. And pi is actually a simple number. I can write a short algorithm to describe pi, but it looks entirely random. It's Borel random, which means that every kind of every sequence and every combination of sequences appears equally likely. And so, because we never know whether there might be some short algorithm that generates this sequence, we say that's formally that's an intuitive way of realizing that it's uncomputable. And this AIT gives us new intuitions. So one of the intuitions is that the formal definitions of a random number is a number for which its Kolmogorov complexity is equal to or slightly larger than its length. In other words, the only program that will generate that number is print the number. And that's a really deep definition of randomness, because it tells you there's no other simpler description that will generate that sequence. Other kind of interesting intuitions are that the complexity of a set can be much less than the complexity of elements of the set. So for example, if I say, write down all numbers from 1 to Googleplex, that's a relatively short description. However, individual numbers in that set could be relatively complex. So you see that you get some kind of intuitions that are not the kind that you're probably used to in your day-to-day life. And this is why AIT is a very deep and extremely powerful set of ideas, which has been languished a little bit in the physics community. And I think that is something I'm hoping to change over the next few years. And how does this link to questions of how nature finds these particular subset of sequences in this very vast set of possibilities? Well, there I can give you another intuition, which I think explains intuitively what's happening, and then I'll formalize it. So let's say you've got a monkey typing on a word processor. I used to say monkeys on typewriters, but of course you guys don't know what typewriters are anymore. So a word processor, then the inputs are the outputs. So how likely is a monkey to type, say, the first m digits of pi? Well, if there's n keys, say 50 keys, it's 1 over 50 to the power n plus 1, because you have to get 3.14, right? So 3.14, you need to have 4 things right, because you have to get the dot in there. So the fact is the probability is low, but actually is equal to any other sequence of that length. However, what if instead the monkey was typing into C? There's in fact a competition to write the shortest C program that will generate pi. And here's, as far as I know, one of the shortest ones by Dick Winter in Amsterdam. And it's 133 characters, and it will generate the first 15,000 digits of pi using a spigot algorithm. So the monkeys, if they're typing, rather than typing into a word processor, they're typing into a C computer, they might accidentally type their 133 characters of the C code, and then they would get all 15,000 digits of pi correct. And this tells you that numbers with sequences with low Kolmogorov complexity are going to be much more likely to emerge when somebody randomly types into a computer program than complex ones for whom there's no program shorter than just print the number. It turns out that the vast majority of sequences are complex. You can prove that most of them are complex, so there is no shorter program than just typing them. So there's only a small number of these ones that are simple. And so if what you're looking at is a process that's giving random inputs, but that input is going into something which is structured, which interprets it in some way, the way a computer program does, then what you're going to see as outputs is simple outputs with short descriptions appearing exponentially more likely than longer outputs. So that's a fascinating kind of intuition, and that intuition was formalized by actually the first person who came up with the idea of Kolmogorov complexity, although he's not been recognized as such because he didn't quite realize that's what he had done. But Ray Solomonoff was thinking in the early 1960s about what happens if you randomly type into a universal Turing machine. How likely are you to get a certain output X? So how likely am I to get a certain output X if I randomly type into a universal Turing machine? For simplicity, I'm going to have a Turing machine that only takes binary code, so 0, 1. Well, how likely am I to get, say, pi? Well, it's going to be the sum over all codes that give me pi. And how likely am I to get that particular code? Well, it's 1/2 to the length of the code. So sum over all codes that give me pi times 1/2 to the power of the length. That's going to be the likelihood that I get that output. The first term in that series is, of course, the shortest program, which is the Kolmogorov complexity by definition. And it's actually the biggest term. So intuitively, he's trying to formalize the idea that simpler outputs are more likely to appear than longer ones. And Marvin Minsky, one of the great fathers of AI, said, it seems to me, not so long ago, that he said this, it seems to me that the most important discovery since Gödel was a discovery by Chaitin, Solomonoff, and Kolmogorov of the concept called algorithmic probability. Everybody should learn all about it and spend the rest of their lives working on it. So what is this idea of algorithmic probability? I just explained it to you. It's as simple as that. It's how likely is a computer to generate something when you randomly type into it. And there's a really beautiful theorem by another great Russian genius, Levin, also the person who came up with the idea of P and NP in computer P and NP in computer science. He pointed out that this is a lower bound of probability because you have at least one code that will generate the outputs, but it's also up to order 1 terms, up to terms that are independent of the sequence, also an upper bound. And that's a very, very, very deep and very subtle and complex idea. But it tells you that basically this monkey intuition that you're more likely to output simple things, simple short descriptions, long descriptions, should hold for a wide range of systems. Now, why have you not been taught this in your undergraduate degree? Well, it's because a number of things. One is that most systems that we look at are not really universal Turing machines. They're something much simpler. This Kolmogorov complexity is formally incomputable, and mathematicians are often interested in that particular property. And this theorem only really holds in the asymptotic limits. It can be proven in the asymptotic limits of very large sequences. But nevertheless, I had another DPhil student whose job was to try to understand why these RNA systems were biased the way they are. And he helped me find a much simpler argument, which I won't sketch here. I won't give you the proof sketch, but it's basically based on the idea that the description of a set of all things can be much shorter than individual elements of the set. So the description of a set can be very simple. And we use that to prove an upper bound on the outputs of a wide range of input-output maps. And we said basically they're going to be 1/2 to some approximation of the Kolmogorov complexity, so not the real one, which we can never exactly give, but we can bound it by various approximations. And this theorem works really well as long as the map that we're looking at is simple. That means that the map itself is not super complicated. You can imagine if you had a very complicated map, then you feed it inputs, then the outputs will not be linked to the complexity of the outputs, but linked to some complexity of the map. So it has to be the map has to be asymptotically simple. And this thing works extremely well. In fact, we applied it to these RNA structures. So I told you, remember, that you could take these structures and abstract them as binary strings. So you can then work out something very simple, which is how compressible is a string. Use a compressor like the one that you use on your computer to compress a document. And this complexity that we use here is basically what's the shortest description I can make on a compressor of the sequence. Well, these very simple structures have very short, low complexities. And this complicated structure has a high complexity. And here you see the probability of getting a particular structure versus the complexity of the structure. And what you see, this is our bound that we predict. And what it tells you is that the log of the probability scales in this way. So this is many orders of magnitude in difference in how likely you are to get a certain output. And so complicated structures like this one are much less likely to appear than simpler structures like this one, for example. And so the kind of cool thing is that I can tell you a priori what RNA structures are going to be likely to appear and which ones are unlikely to appear. And that's just based on how simple their descriptions are, which is simple means how compressible they are. So this is interesting because I apply this now to the library of Babel, Borges's library of Babel. This tells me that the more compressible my story is, the more I can compress the book, the more likely that story is to appear, the more ways that story can be told. And so I'll predict that if you were to look at that library at all possible stories, then you would find that very highly compressible stories are the stories that appear the most exponentially more likely than less compressible stories. So that's basically the story there in evolution. And we did a wide range of different systems of coupled ordinary differential equations. This L systems here is actually a plant. It was originally done for plant biology, but it's now used in computer graphics. So if you ever watched this old movie called Avatar, I think it's still the highest grossing movie of all time. And it's about a fantasy world, a kind of outer space with this beautiful, lush tropical scenery. And that was all generated using L systems. And so if you take L systems, which are little algorithmic ways to making plant-like shapes, it turns out if you randomly make them, you're much more likely to make simple plants than complicated plants. And you can predict how much more likely you are in this exponential way by this bound. And this explains a lot of the structure that you see in that system as well. Let me see how I'm doing on time. So I'll give you one more example of this, which I think is kind of cool. So this is actually how I got interested in self-assembly. So this is a little movie here of the bacterial flagellar motor. So bacteria swim. It's a little motor that spins a flagella behind it. And what's kind of cool about this thing is that it self-assembles. So each of these little things is a protein which spontaneously forms into this very well-defined shape. And that makes the motor that turns the flagellum around. There's lots of interesting questions about how this thing self-assembles, which got me interested in self-assembly. But I was also got interested in how does this thing evolve? Because evolution has to randomly change the way these proteins bind with each other and somehow come up with this very well-structured object. How does it do so? So we started working on a typical physicist model of proteins, little squares that have sides that can bind to one another. And so like this protein will be made of two components, come together in an 8-mer will become this structure right here. And then these things are actually called polyominoes because they're like dominoes, 2, 3, 4. It's a branch of recreational mathematics. And so you can do questions like this. If I get you this structure, then I can ask myself, can I make a set of particles that have the following set of rules? I put the first particle down, and then the other ones just come in and randomly stick or don't stick. Okay, it's a little bit like those proteins floating around. Can I come up with a set of rules where they stick always into this particular shape? Well, for this set of rules and this set of blocks, that will happen. This first one comes down with little letters A on it. A, in this set of rules, if there's a 1, it binds, a 0, it doesn't bind. So A only binds to C. 3 is the only one with C, the 3's bind there. C has D's on the side, D's bind to B's. The only one with a B on is number 2, so these 2's bind, etc. And I think this continues on, and this structure forms each time. It's a little bit like that self-assembly problem I showed you with the flagellar motor. It always forms in exactly the same shape. So that's the design question. I have this set of blocks, set of rules, I get this structure. But now I have to turn this question around and say, if I give you this shape, can I find a set of rules that would make it? Well, you can do it by thinking about it and playing with it. You can use graph theoretical methods, but you can also just use an evolutionary algorithm where you basically set up a random set of blocks and rules, see what it makes, pick the ones of the set of rules that you've made that's closest to this your target structure, and then mutate those, and then pick the best ones out and mutate those, and keep doing this over time. And eventually you'll find a set of rules that will generate this. And so my DPhil student, Ian, was playing with this and said, well, let's make 16 MERS. Okay, so 16 different blocks, so 16 ominoes. It turns out that there are 13,079,255 different 16 ominoes. So you might think that if my fitness function doesn't say make a particular structure, but just make a 16 MER, then I might get each of those 16 MERs with equal probability. But what Ian found when he ran the evolutionary algorithm is that actually this small set of 21 shapes appears about 50% of the time. And what's even cooler about them is this small shape, which are the ones that appear half the time. So they're they're almost 1 million times more likely to appear than the average polynomial is. They also include all the symmetric polynomials, the highly symmetric ones. So there are in that set of 13 million, only 5 that have what we call D4 symmetry. It's a symmetry of the square. You can flip them, rotate them, and they themselves. And they're actually in that little small set of 21. So they appear almost they appear like for free. You get the highly symmetric ones. And the reason for that is because the probability complexity argument that I gave you for holds for these things. This is the bound that we predicted. And so these symmetric ones obviously have low description lengths, because I just need to say, here's something that I repeat multiple times. That's a short description, as opposed to a complex structure like this one, where every single particle might have to be slightly different. So the description is long. And so simple structures, symmetric structures, have short descriptions and are therefore much more likely to appear in nature. And we've looked at real protein clusters, so proteins that are found in nature, and they're hugely more symmetric than you would expect just naively. And that symmetry, we argue, is based on the idea that symmetric things are just easier to find. And that's kind of cool. So symmetry spontaneously emerges from this algorithmic picture that I just gave you. So here's a little example of the protein structures. I won't bore you with that too much. The last thing I want to do in the last, say, 5-10 minutes is see whether these ideas can be broadened outside of evolution into machine learning. So machine learning is has become extremely popular even in physics now, and it's basically where it's some kind of system where you try to train a machine, try to train a computer program to do something by giving it lots of examples. If you're doing supervised learning, so for example, I might give you all these images, and I've got a neural network which has some inputs, which are the images. It's got a bunch of weights that I can vary, and it has outputs that tell me whether hopefully tell me whether the image is a cat or a dog, or a pool, or a car, for example. And so what you do is you take a bunch of these images and you train, which means you feed it into your network, you fiddle with your weights until it gives you the correct labels on the things. And then once you've got it all trained, then you test it on some new images that you haven't seen. And if it does well on the new images, then say it generalizes well. So if it sees something it hasn't seen before, but it recognizes it, then you see it generalize as well. And one of the kind of conundrums of the neural networks is that they're hugely over-parameterized. So they've got typically many, many more parameters than the data points that you're trying to learn. And that's surprising, because we've been taught in physics from a young age that if we put too many parameters into a system, it won't work. So very famously, John von Neumann said to Enrico Fermi, with 4 parameters I can fit an elephant, and with 5 I can make a wiggle its tail. So we're taught if you have too many parameters, things don't work well. Here's an example. If I give you these data points here, these 10 data points, and you fit a 5th order polynomial to it, you get a reasonable fit. It won't get all the data exactly correct, but you won't be that far off in between. So you might generalize not too badly on data points that you don't know, that XY pairs you don't know yet. If I do a 20 order fit, I can fit data extremely well, but I can get these huge oscillations because it's typically a complex polynomial. And it's intuitively, we say this can't not possibly be right, because you don't expect to have that kind of fluctuations in your real system. Interesting, a neural network, even a neural network with thousands and thousands of parameters, they all fit with this very, very smooth function. So the question is, why do neural networks, given that they can actually fit any polynomial, in fact, you can prove that they're perfect function approximators. They could fit any polynomial, any other function form to your data. Why do they pick this very small subset of simple ones? And for that, I want to give you an even simpler example, to give you some intuition for how this might work. So I'm going to work on a Boolean system, which basically is this. It's a doctor's decision table, say for COVID-19. So let's say I want to train a machine so that if you come to it with some symptoms, it knows whether to send you to the hospital or not. So you have a fever and you're coughing, you lost your sense of smell, you're over 50, you've got a heart problem, you're obese, you have diabetes, then I might send you to the hospital to be checked out. If you have those symptoms, but you're younger, but you also have some comorbidities, I might still send you to the hospital. If you have the symptoms, but you're young and otherwise healthy, I might say to you, well, just tough, stick it out at home, etc. So you might think, well, this is relatively simple. I've got 7 questions. So I've got 7 questions. So I've got each one that I can say yes or no. So that's 2 to the 7 possible answers, 120 possible answers. How many different functions do I have? So a function is whether I send you to the hospital or not. Well, it turns out that because for every set of answers, I can give you a different send you to the hospital or not. There's actually 2 to the 128 possible functions. So 10 to the 38 possible functions. And the neural network could pick any of those functions to fit the data. What we find in practice is that it actually does a really good job at predicting in the future what the answer is going to be. And you can kind of see it because there's structure to this data, right? So clearly, if you're young and otherwise healthy, is very different when you're old and not very healthy. And so those are patterns that are in there. But how does the system know about these patterns? Is the kind of question that you might ask. And so what we've done is we've looked at this as a mapping. This is a mapping from functions, from sort of parameters to functions. So each of these different outputs is a different function. The parameter of the neural network encodes those functions. And I can ask myself, if I randomly pick parameters, how likely will I get a particular function? Okay, that is very equivalent to this question I've been saying again and again. What if I randomly pick genotypes in an evolutionary system? How likely am I to get a particular outcome? And what we find, not surprisingly, is that of all the functions, a small number of functions are very likely to appear. So this is the probability to get a function versus their complexity. Simple functions are likely to appear and complex functions are unlikely to appear. So in this case, even though there are 10 to 38 functions, the probability of getting, if you just chose functions at random, you'd have them with a 10 to the 38 chance, 1 in 10 to 38 chance that you would get the same function twice. But if you pick the functions by randomly picking parameters, then you're going to find a small subset of functions that are much more likely to appear than others. And this, we think, explains why neural networks generalize so well. So why when you train them on something, do they pick functions, predict very well for new data? That's because I'm showing you here is the accuracy of the neural network after I've trained it on a simple target function. So a function here, which has a load, which is highly compressible. If I train it on one of those, and then I look for more data that I haven't seen yet, so I train it on half the data. So for example, let's say I gave you half the 164 of the answers. I said, can you predict the other 64 of the answers? If this is a very simple output, so for example, the output's all zeros, that's very simple, then it will learn it really well and predict that you're going to keep getting zeros. Or if it has another structure to it, it's going to pick that up and predict it. Whereas if the function you're trying to learn is very complicated, then the neural network won't do well. On the other hand, if I have an unbiased learner, so a learner that doesn't use this neural network system, but just picks different functions at random that fit the data, then the vast majority of functions that fit the data give you very bad predictions, very bad generalization. And so this neural network has an inbuilt Occam's razor towards simple structures. And that's really helpful because simple outputs, we think, describe a lot of what happens in nature. So if you look at images, they're very, very far from random images, which would be complex. They're actually quite structured. And so having a bias towards simple structures is a really, really good thing to have. And we think, so our argument is that this same algorithmic information theory argument, the monkey on typewriter's intuition, can be applied to neural networks, where randomly picking parameters in the neural network is a little bit like a monkey randomly typing on a keyboard, but you're not randomly picking functions. Those random parameters are then processed by the neural network to pick functions, and the neural network is intrinsically biased towards simple outputs, as we just showed in the system, and as we've shown much more generally for a wide range of other neural network systems. And this bias towards simplicity happens to be a good bias. It could have been a bad bias, in which case we would see bad predictions. But it's a good bias because, in fact, the stuff we look at that we're interested in is typically simple. So I hope I showed you a little bit about information theory, but not the information theory you might be used to, Shannon information theory, but rather algorithmic information theory. And I use that to explain how RNA, why certain RNA structures found in nature are much more likely to be found, and those are the structures that have that are highly compressible. I made an argument for why this works for neural networks, why certain functions are much more likely to be than others, and this is also a good thing. And then for these self-assembling proteins, I use the same argument. And the kind of fascinating thing is that this argument, this coding theorem from Levin that we simplified into our little upper bound, works for this wide range of non-universal systems, which suggests that it's something that we might see much more widely across the physical world. And with that, I want to thank you for your attention, and I'm happy to take some questions.
Thank you very much, Professor. We've got a couple of questions coming in already. Everyone else, there's 2 questions now, ask your questions as well. Let me get started until people think of some questions. So my question is the whole argument about, the simplicity bias.
Yeah.
It does have predictive power. And we've seen that by, predicting the RNA structures and all that should come up. Has it been used in a case where we don't actually know the structures that we find and get what structures we expect to find?
That's a good question. So so far we've just used structures that are found in nature.
Right. Yeah.
And what we said is, here's the structure we expect to find, and the structures found in nature are the ones that we expect to find. What we are hoping to do fairly soon is to write a paper and saying, and if you keep looking, okay, you're going to find these structures, but not those structures. So in the case of the, say, length 55 RNAs, where there's 10 million possible coarse grain structures, to be easier to work with, we find that there's 28 that are used by nature out of the 10 million. And I can say with pretty much confidence that you're going to, if I could give you the top 50, that's all you're going to find in nature. And I can tell you how likely you're going to find them in nature as well. That's the kind of prediction that we have.
Next question by Jeremy. Do you think this kind of analysis relates to the general appeal of symmetry and simplicity in art and music, etc.? Could somehow this bias towards certain configurations be encoded into our minds?
Well, this is I actually have a DPhil student coming next year. She's going to be working with me on neuroscience because I'd like to learn more about it, because I have a very strong intuition that our minds have these kinds of biases in them as well. I step in this with some trepidation, because physicists are famous for stepping into fields and giving wild, easy, simple predictions without properly understanding them. And that's a field which is vast and has lots of very smart people thinking about it. So I've got a very smart DPhil student, and she's going to teach me some basic neuroscience. And after I've understood it better, then I will see whether my intuition is nonsense, in which case I'll slowly try to do something else. Or if it's interesting, I'll pursue it and try to get more funding on it. Like, this is one of the things I like to do. So I for my MPhys projects and for my DPhil projects, I often choose topics that I don't know very much about, so that students can teach me something. And so the RNA was something that my student Stefan taught me initially, and other students have gone on since. And in fact, I just had a very bright MPhys student, Tasmin Sarkani, do a very beautiful little piece of work on RNA that I learned a lot from for MPhys thesis. And so each of these cases, and so the neuroscience is something I'm very interested in. Now, clearly, our minds have all kinds of biases. And one of the very famous questions in the field has been, for example, language acquisition. So children acquire an ability to understand grammar very quickly. And famously, Chomsky pointed out that this seems to be too fast, given the amount of data that they've been given. So there must be some kind of intrinsic bias. He calls this universal grammar. This has been hotly debated for a long time. And it might be that these kinds of ideas will help explain some of those patterns. That's the physicist. That's the thing I'll say to you physicists. I won't say this to my neuroscience friends, because I will sound like another physicist about stuff they don't really know about, and that's probably fair. I think amongst ourselves, we can speculate on these things, and Who knows?
Yeah. Next question, Oscar. Love number of Physics Society. Oscar says, "Hi, Professor. Thank you for the fascinating talk. My question is, how useful do you expect quantum computing to be in your field? Are you currently limited by the computing power when running the algorithms?" That's the first question.
Yeah, so the kinds of algorithms we're running are not ones typically that for which quantum computing makes a huge speed up, but I could be wrong. So I'm not expecting quantum computing to solve these problems very well, but there are some very fascinating connections between algorithmic information theory and quantum computing that I think are well worth working out in more detail. That's probably as far as I can go, but I don't. So a lot of the quantum computing work on information theory is general information, and I'm looking at something slightly different, which is algorithmic information, and it's a different kind of beast, although there are deep connections between them.
Then, Oscar again, you have made it seem like all these protein structures seem essentially inevitable. Has your research changed your view on the probability of life of any kind on another planet?
Well, no, I actually have spent a lot of time thinking about this, and I mentioned my friend Simon Corey-Morris in Cambridge, who's a very, very interesting paleontologist, and I actually ran a conference with him. We looked at some of these questions, and so I'm very interested in it. I think questions about the origin of life are very complex and often harder than they look. Nevertheless, I think the following is true, which is that with a relatively small number of random samples of sequences, I'm able to generate all of the structural diversity that you see at this coarse-grained level. Okay, so not all the structural diversity you see in nature, but all the structural diversity you see at this coarse-grained level, so that the basic structures are there. That's very promising, because that suggests that you can get enough structure to allow life to occur with a relatively small amount of random sampling. And so the question of these hyper-astronomically large search spaces has been, it's actually a long set of conflicts between mathematicians and physicists and evolutionary biologists. It's a very fascinating set of conferences that was run in the 1960s called the Wistar conferences. They brought physicists, mathematicians, and biologists together. And there's a very famous French mathematician called Schlumberger, who is also very important in AIT. And there's an absolutely fascinating exchange in that they actually typed everything out, so all the exchanges were transcribed. And he basically says, look, these spaces are very large. He works out how big these possibility spaces are, how hard it is to find anything in those spaces, and says, well, how does evolution work that it finds things in these high spaces? And the biologists basically say, well, I don't see why it's a problem, because clearly it has. So what's the problem? And that's a fair answer, but not a satisfying one for him and not a satisfying one for me, because I want to understand why. And there's actually a fairly established trope in kind of anti-evolutionary literature that repeats the same argument. It says these spaces are too big to be searched, ergo evolution can't have found solutions in them. And I think that the work that we're doing is showing that even though it's true the spaces are very large, that you don't need to search all the way through them, that you're going to keep finding the same small subset of things each time. So I wrote that, I had a little link to the paper on hyperastronomical numbers and convergence and contingency in evolution. So contingency is the idea that you're going to find something different every time. Convergence is the idea that you find the same thing multiple times. And so my arguments are basically for a certain type of convergence. And I'm claiming that the coding theorem, this AIT, explains the kind of convergence that you're going to see. So it's scratching at the surface of a very complicated problem, but it's pointing very, very much towards a solution of some kind for it, even if I think we're still far away from something definitive.
Very interesting. Thank you very much.
And we have 4 more questions. So Harry asks, what's the link between universal Turing machines and biological systems?
Yes, exactly. So so universal Turing machines are basically computers that can do any possible calculation. So you typically have a hierarchy famously Chomsky hierarchy, or the hierarchies of computers from very simple finite state machines to universal Turing machines. You can do anything that can be computed by a universal Turing machine. So there are people that have looked for universal Turing behavior in biological systems, and there's some claims that might be found. What we're arguing in our papers is that you can see a lot of the same behavior in non-universal maps. So systems that are not universal Turing machines. So biological systems, like the RNA system, is not universal Turing machine. It's a certain type of, it's somewhere in the middle of this hierarchy of computational complexity, but of complexity of machines. But it nevertheless shows behavior that you can only formally prove for universal Turing machines. So what we're actually saying is that we think the question of whether or not biological systems are universal is perhaps not as interesting as people may have thought, because we can reproduce some of the really interesting behavior of being universal.
Mm-hmm. Oh, that's very cool. Carol, very interesting talk. Thanks a lot. I wonder how your result that self-assembly has bias towards low complexity relates to the second law of thermodynamics. You mentioned the Shannon entropy measure. Assuming that the second law is formulated in terms of Shannon entropy, how does Kolmogorov complexity relate to Shannon? And then, is your main result of low complexity bias still compatible with the second law?
Yeah, so the second law is, and these are two kind of slightly different things. The second law you might think of as working in the space of the sequences, right? So if I randomly pick sequences, I'm much more likely to get a complex sequence than I am to get a simple sequence. But the outputs are a different thing. It's a question of how likely certain things are to appear than others. And so there's a bias to that system. Just like if you think about the second law of thermodynamics, it's still the case. In fact, it's the case that certain macro states are much more likely to appear than other macro states because they have larger entropy or less entropy. So the question of the link between Kolmogorov complexity and Shannon entropy is a very deep and subtle one. To first order, I gave you that example of these simple sequences like 0, 1 many times or a random sequence. The simplest Shannon entropy of both those is the same because they have zeros and ones with roughly equal probability. So they're both high entropy sequences. But clearly that's binary entropy, but I could make entropy, which is a block entropy. So entropy of 0, 1s. And then suddenly the entropy is very low because that would be like 0, 1 repeats again and again and again. That would be like a coin which always comes up heads if I consider 0, 1 to be a head. And so you can use block entropies to approximate Kolmogorov complexity, which is effectively what we do, a sophisticated version of block entropy. And then you can prove a bunch of things about links between Kolmogorov complexity of a sequence and the Shannon entropy of it, where you treat the sequence as if it's being produced by a stochastic process. And that's actually, I got a DPhil student working on those questions at the moment. I'm saying anything. I'm not making any kind of claim that what I'm doing is overturning the second law of thermodynamics at all. What I'm basically arguing is that when you're looking at, say, self-assembling systems, then you're much more likely to get symmetric structures, for example, or simple structures with simple descriptions than complex ones. That's a question about outputs like states rather than the microstates. So basically I'm saying structures that have simple descriptions have many more microstates that map to them than structures that have complex descriptions.
Then NH asks, have you investigated the role of evolution mutations in transitioning between different simple states?
That's an interesting question. So maybe, I wonder if you could expand a little bit on the question.
Yeah, NH, if you're still with us, feel free to expand.
So maybe I can say this. So actually, maybe I think what you might be answering is this question, which I think is a very interesting one. So if I have a certain RNA structure and I randomly mutate it, okay, what is it going to mutate into? Well, we've actually worked on that problem, and what we show is the structures that are much more likely to appear by random sequence picking. That's not really how evolution works, right? Evolution doesn't pick sequence random. It starts from a particular structure and then mutates that. but the likelihood that you get let's say I've got structure A has many sequence mapping to it, structure B has very few. If I have a structure C and I mutate it, I'm much more likely to get A than I'm likely to get B. And the likelihood that I get A is proportional to the number of sequences that map to A. And so that's why I get A much more likely than B. So what I'm just basically saying is so we have looked at evolutionary transition. So if you take a sequence, you start mutating it, where does it go? And it basically is much more likely to find simple structures than complex structures.
Next question, Zina asks, thanks for the amazing talk. What is your view on consciousness? Do you think it is computable if it exists?
Well, I don't have any strong ideas about consciousness. I think it's the hard problem, as people sometimes say. There is a famous argument by a philosopher called Lucas here in Oxford, who's now retired, but it's a famous argument where he claims that consciousness is uncomputable by mapping it onto these questions of halting problems and Turing machines. Not everybody's convinced by the argument, but it's an interesting and intriguing one. So that would be my only thing I would say is if you want to make this link between computability and consciousness, look up Lucas consciousness and computability, and you will see a very famous paper and then a very long history of people who have interacted with that question and either agreed or disagreed with him. But nobody said it's uninteresting. So.
And the last question looks like Vasilis asks, thank you for the very interesting talk. What is your take on the relationship between self-replication and the observation on the commonality of the observed sequences?
Let me think if I understand the question properly. I think there's a deep question behind there. So so here's one way of thinking about this. So self-replication is basically when you make copies of yourself. And one of the really interesting things that we've looked at, actually, my student Tasman was looking at this very recently. So I just said to you that if I mutate it to find a new structure, then the likelihood that I find that new structure is roughly proportional to how likely that structure is going to appear de novo by just randomly picking sequences. That's not true for how likely I am if I mutate to find myself. It turns out the likelihood that I find myself again, so that I am, in other words, the likelihood that I am robust is much, much, much higher, is exponentially higher. And we're trying to, so we've basically shown this is true, and we're currently working on some proofs to try to prove that this is why this holds. And there's some really cool stuff linking to something called bricklayer graphs. And it turns out actually, if you work out the maximum robustness of like these RNA structures, that the maximum robustness can be linked to a function called Tagaki function, which is everywhere continuous but nowhere differentiable. And this is just a curiosity that appears. I don't think anything particularly deep about it, but it's kind of fun. And so you're much more likely when you self-replicate, therefore, to be yourself again, which is a good thing, than you are to be something else, because you want to have a certain amount of robustness, so that basically, if you're very fragile, so every time you mutate, so your offspring are completely different from yourself, then eventually you're going to fall off whatever fitness peak you're on. And so we actually think that this will, in fact, I have another very bright student, Vaibhav Monty, who's basically, we think we've shown that there's a phase transition in the mappings from sequences to structures, that you go from either a kind of completely mean field like random map to these correlated maps, which are the ones that we see in nature. So we're looking at this as a phase transition between two states. So it's kind of cool. Lots of physics. Physics is appearing all kinds of different ways. Information systems. Yeah, it's really cool.
And NH elaborates on their question. So NH asked the role of evolution in transitioning between simple states, and now say they were wondering if mutations from one simple state A could lead to another simple state B.
Yeah, so I think the answer is that actually what we think is the case is that the likelihood that you get state A or state B is basically directly proportional to roughly how simple they are. The simpler they are, the more likely you are to
Right.
get a map to them. And this is more generally the case. So there's a surprising thing there that we don't really understand, which is that this would suggest that if you keep mutating, you're going to go towards simple structures. Evolution has clearly become more complex over time. And so the question is, how does that work? And we think the argument goes a little bit like this, which is that these structures that are simple, so they have many sequence mapping to them, are also more robust. That means that they're less likely to break when you mutate them. And that means that if you're very robust, that means you can start tweaking part of your sequence to add new functionality. So for example, if you're one protein, you can tweak your sequence so that you can stick to another protein without actually messing up what you're doing as a single protein. And so we think that actually allows you to then form modular wholes. And we think that must be a very important part of how biological complexity arises. But this is now very much hand waving.
And yeah, I think this was the last question. Everyone seems to have really enjoyed the talk, myself included. I thought it was quite profound, I think. So yeah, I guess this is it. Everyone, thank you very much, Professor Ard, for like, you know, explaining to us all these ideas.
Well, thank you. I really enjoyed my chance to speak to you. And thank you very much for listening and for all the great questions.
Absolutely. Yeah, so thank you very much for coming, everyone. And yeah, thank you, Professor Ard Louis.
Thank you. Bye.
Bye.